Okay, we can get started here. Welcome to this week's colloquium. Matt Kopinski is traveling, so he asked me to introduce the speaker this week. It's a great pleasure for me to introduce Professor Gmitro. He's the Fenton Maynard Chair in Cancer Imaging and Professor and Associate Vice Chairman for Research in the Department of Medical Imaging in the College of Medicine. He's also one of our distinguished alumni. And he has a joint appointment in the College of Optical Sciences and is an active member of the Graduate Interdisciplinary Program in Biomedical Engineering. He is also the co-leader of the University of Arizona Cancer Imaging Program. Professor Gmitro got his PhD in optical science in 1982. No, not 1982, yep. And
couldn't be that long ago, could it?
Under the mentorship of Professor Harry Barrett. And he was the assistant professor of diagnostic radiology at Yale University after that, before he joined the University of Arizona faculty in 1987. So he has received multiple awards, including the Rudolph King Slate Award from SPIE and the Francois Erbsman Prize from IPMI. And so, he's also mentored 20 doctoral and 4 postdoctoral students. And he today is going to tell us about optics and image science in cancer research and clinical care. So let's welcome Professor Gmitro.
Thank you. Well, it's quite an honor to be here. Long ago, I was one of the students sitting in the audience. I can't remember quite as long as that, but it is an honor to be back and be able to present here at Optical Sciences. I wanted to do, I guess that's a fairly broad title for my talk, and so I don't think I can cover that range of material, but I do want to give a talk that covers 3 basic areas that all relate to that. And one is, as co-leader of the Cancer Imaging Program, I want to give a little bit of background about what that is. Many people over on main campus are less familiar with some of the activities in the medical school, so I want to spend a few minutes talking about the Cancer Imaging Program, and then go on to a few projects, a couple of specific projects in my laboratory, one of which is aimed at trying to develop a better diagnostic tool that can actually get in and assist, help patients, cancer patients, in this case, primarily we're looking at ovarian cancer, and then another project that we're developing as a basic tool for cancer biology researchers. So that's the aim or the outline of the talk. So what is the Cancer Imaging Program? Well, the University of Arizona Cancer Center is a comprehensive cancer center, one of about 30 in the country. It has 4 main scientific programs: cancer prevention and control, cancer therapeutics development, cancer biology, and the Cancer Imaging Program. The major aims of the Cancer Imaging Program are to try to advance fundamental knowledge in image science. And this department knows very well what that means. Many people don't quite understand the concept of image science, but Harry Barrett and others have really pushed that forward in a very solid way within this college, and trying to bring that kind of approach to cancer. So that's one of our main goals. Another one is to develop advanced imaging tools for studies in basic cancer biology. So I'll be talking about one project there. To improve diagnosis through development of imaging instruments, techniques, and methods. Again, try to touch on that. And then finally, to improve cancer therapy through development of imaging approaches to predict and or monitor therapeutic response. This is actually an area, this last one is actually an area where a lot of work is going on to try to use imaging techniques to predict very early on whether patients are responding to chemotherapy or any other kind of therapy, and modify or improve the therapy so that they do respond. So it's a very active area. There are a number of connections between the Cancer Imaging Program and College of Optical Sciences. Harry Barrett, Lars Fernlid, Matt Kopinski are all part of the Center for Gamma Ray Imaging, working on nuclear imaging as well as the image science aspects broadly across all imaging modalities. Jennifer Barton gave a very nice colloquium a few weeks ago, who's an expert in the area of OCT. Her laboratory's done some pioneering work there in the applications of OCT, endoscopic OCT for cancer applications. Ray Kostick, as many of you know, is developing a unique holographic approach to multiplanar imaging in thick biological samples with cancer applications. Ron Liang is working on a parallelized OCT system. Lele Pang has some very unique approaches to microscopy, advanced microscopy methods in fluorescence lifetime, as well as instead nonlinear type approaches. Andy Rouse is a faculty member who works very closely with me, and you'll see some of his work today. Urs Utzinger is a member in BME and joint with Optical Sciences. Does a lot of work in optical spectroscopy as it applies to cancer, as well as nonlinear imaging. And Russ Whitty is somebody that's been working for a while now on photoacoustic imaging. So there's a lot of connections, and I encourage people who are interested in this area to contact us and see if you would like to get involved. Final thing is, what does it do for you? Why would you bother being part of a cancer imaging program? Well, I think the biggest thing is it allows close connection and interactions with a whole community of clinicians and basic scientists that are working very diligently. Their careers are dedicated to cancer research and improving cancer care. So there's a whole community over there, and many of us work with people at the forefront and can make connections for those who are interested in moving in their research in that direction. It gives you access to the University of Arizona Cancer Center core services, the shared experimental mouse, the tissue acquisition, cellular molecular analysis, shared service, bioinformatics, clinical trials. All of those things are core services that help people get research done in this area if you're starting to move towards human and or animal imaging. Part of the regulatory process of working with humans or animals to help get you through the bureaucracy there. And then there's a number of very interesting seminars. We hold a monthly CIP meeting with lunch catered from forget, Baggins? No. Yeah, Baggins. So that's kind of fun. And if anybody's interested, get email to me, and I'll get you on the listserv, and you can keep track of what's going on and come by and see some of the interesting talks that are presented. So with that, I'll switch gears, unless anybody has any quick questions about the cancer imaging program. No? Okay. Well, catch me afterwards if so. And I'll talk now about two specific projects that have been going on in my laboratory for some time. One is confocal microendoscopy. So typically, if someone wants to do a diagnosis of disease, they have to go through, or a typical way of doing things is through what is called conventional biopsy. You take tissue out. You have to extract tissue. So what does that mean? Well, this is an example here of a video of showing an endoscope. So there's another endoscope looking at this. I think this is in an esophagus. So you can see this endoscope moving around tissue, and there's a port in that endoscope through which you can stick a tool to extract tissue. So this is a grabber that can go in and grab a piece of tissue and rip it out. So that's what you need to do to extract tissue in many cases. There's lots of other ways to do it through needles and other things like that. It looks worse than it is. People heal up. But after that, there's a whole process. It's not just extracting tissue. You've got to fix it in formalin, embed it in paraffin, cut it thin slice, mount it on a slide, stain it, put a cover slip in it, and then finally you have something, a slide that you can take over and give to a pathologist to look at. So the idea is then you take the tissue with all this preparation and time to the microscope and have an expert read it and make a diagnosis. What optical biopsy is trying to do is don't bring the tissue to the microscope, bring the microscope to the tissue. So here's an example of in vivo imaging where there's a big piece of tissue that you'd like to know whether it's diseased or not, and you can bring a specialized microscope into contact and get an image from which diagnosis can be made. The advantage, less invasive, you don't have to rip tissue out, more coverage, you can move this around quite easily and scan around, get a lot more information, less error therefore because of what's called sampling error. You might have taken tissue from the wrong location, and you get real-time feedback. So there's a lot of advantages to techniques and technologies that can produce optical biopsies. And so the approach we're taking is one of many. Jennifer Barton gave a very nice description of OCT in this context of optical biopsy, and there's nonlinear microscopy. There's lots of ways of approaching this. But what do you need for all of these? For in vivo optical biopsy, you need to be able to essentially section, like this thin section of prepared tissue, a 3D object without actually cutting it. So how do you do optical sectioning? You want to do it rapidly to freeze motion. As you saw, that endoscope was moving all over the place as the patient was breathing and whatnot. So it needs to be fast to freeze motion. You would like good contrast to be able to make a diagnosis. And you need to be able to access tissue and guide your device to the right spot. Think about a microscope that's imaging a teeny little field of view of less than a millimeter in a big organ like the inside of the body, laparoscopically, or the colon. You have to be able to guide to be able to see where it is you want to be able to look. You want the instrument to be small, robust, sterilizable, and if you're going to use contrast agents, external contrast or exogenous contrast agents, you want them to be safe and to work to provide good contrast. So the approach we've taken is confocal microscopy. So how does a confocal microscope work? There's a point source. It's imaged into a thick 3D sample at a certain location, and mostly what I'll be talking about today is fluorescence. If it fluoresces, there's a lot of light there, and the fluorescence from there gets imaged confocally onto a pinhole aperture. A lot of light gets through from that particular location. But what about other locations, like back here? It's in the illumination beam, so it is fluorescing, but when it images that fluorescence, it's blurred out. So not much light gets through, both because of detection sensitivity and also because it's in a much weaker beam over here, because it's in a spread out beam. Same thing is true on the other side of focus over here. A weaker beam of illumination and a blurred image of that emission that not much gets through. So that's beautiful if you want to image one point. What else do you need to do? Obviously, you need to scan to build up a 2D or 3D image of a 3D sample. So you need to scan laterally as well as in depth if you want to go through the whole sample. So, but what is the advantage here? This is something that produces that optical sectioning, I said. In a big 3D sample, rather than getting one plane in focus and a lot of other planes out of focus, there you get one that is, you get light from the in-focus plane and almost nothing from out-of-focus planes. So you can guess which one is the confocal image as compared to the non-confocal image. Here you have a plane that's in as good a focus as this. There's just a lot of overlapping structures that blur out what you're looking at. So clearly this is a way of doing optical sectioning and one of the big advantages of the approach. So here's a fluorescence confocal microscope, a laser illumination of the tissue, some kind of XY scanning mechanism, usually galvo mirrors that scan the point across the sample. The fluorescent light comes back, is descanned through off of a dichroic mirror that reflects the longer wavelength light to the pinhole aperture. You detect this, and as you scan, you build up a 2D image. Quite straightforward and useful and used all the time in basic biology. So what did we do? We started this, amazingly, 20 years ago, of just taking a confocal microscope and coupling it to a fiber optic imaging bundle, something with, say, 30,000 elements, fibers that are coherently oriented such that an image put in here comes out over there. And you also need some other things, focusing mechanism, ability to deliver dye if you're going to do it. But this is something that was a fairly straightforward adaptation to take a confocal microscope into confocal microendoscope. And there are commercial instruments available to do that. So one of the other requirements was to do things fast. If you want to be able to image in a living system, you want to be able to run at 30 frames a second or faster. So one of the approaches that we took to speed things up was a slit scanning approach. So rather than producing a point of light that scans in 2D, produce a line of light that scans in 1D. So here's a lens system. The illumination looks something like this. It goes through off of the dichroic beam splitter, focuses down off the scan mirror, and is focused onto the input. That would produce a point of light, except for a cylindrical lens here in the other orientation, in the direction out of the plane, blurs that along in the vertical direction, so you get a line illumination rather than a point illumination. And this scan mirror then only has to scan one orientation to scan that line across. The fluorescent light comes back through the dichroic beam splitter, passing it to a slit confocal aperture, and then off of how we do it is through a second mirror that's synchronized to this mirror. So this is static image. The line is scanned across here. It's positioned, shifted, and then it's descanned by the scan mirror to a fixed position of the slit, and then you rescan that to build up a 2D image. So afterwards, you've got a fluorescence confocal microendoscope image of the sample that you were illuminating. This system also has a multispectral capability. This system almost naturally begs as a spectrometer, right? Here you got a slit coming into a system with information along the slit, but you could also disperse the light and get spectral information. So by scanning the mirror off to, this is the grayscale mode of operation. This mirror scans in synchrony with the other scan mirror and builds up a 2D image. You read it out the camera at 30 frames a second and get a real-time view of what's happening. If you want to go to multispectral imaging, go off the mirror, divert the beam over through a prism that's going to disperse it in this direction. In the out of plane or out of board direction is the imaging slit direction. So you have one dimension of spatial information and one dimension of spectral information on a frame. And if you then slowly scan that first mirror across to scan the illumination, reading out multiple frames, you can get the other direction, X. So you get a full 3D data cube of XY lambda, XY lambda. And so in the grayscale mode, you'd get something like this. Let's say in the multispectral mode, you had some different kinds of dye or dyes that have different spectral properties in different environments. You can pull out that through and pseudo-color that to display color information. So there's some advantages to the multispectral mode of operation. The other thing we want to do is be able to focus at varying depths. If you want to build up a 3D image or to be able to image at something deeper than just the surface, you want to be able to focus. So the way that's done is to move the fiber bundle relative to the fixed lens at the end. So if this moves, the conjugates change. You move in, the focal plane changes. So that means that the lens must be well corrected over a broad range of conjugates to be able to do that. That's a challenge. Here's just an example video of looking at a through focus image of a piece of tissue. You can see different cells becoming visible, some brighter than others, and not the greatest example of focus, but it shows. Here's another example, or here's the idea of dye delivery. If you could give a systemic dose of dye, it was a completely safe thing, but you're just trying to image a very localized piece of tissue. So it works better to, go back one and play that one more time. It works better to deliver a very small amount of dye just to the very tip that will impact or touch the tissue and distribute and allow you to image what's going on at that location. So the contrast agents is really an important area. This is something that I think is really going to make or break this field in terms of making it a clinically viable tool. Right now we're just using a dye called acridine orange, which is a vital nuclear stain. It goes across the cell membrane rapidly. It stains the nucleus and gives us very nice quality images. But it's potentially cytotoxic and mutagenic and not something that you want to use generally. So there are some other dyes that are FDA approved that are safer, don't necessarily produce quite as nice a contrast, but there's some choices here in terms of what you might look at for contrast agents for fluorescence imaging. The other thing is that in many diseases have changes in the biochemistry such that there's often changes on the cell surface that are upregulated receptors. For example, the folate receptor is upregulated, overexpressed in ovarian cancer. So you can make an antibody to folate or something that will bind to the folate receptor, a ligand, and attach a fluorophore, any of the safe ones like fluorescein or other things, to that molecule and make a targeted contrast agent that can preferentially go to disease and help you locate and visualize what's happening. That's an important area, but I won't say too much more about it. Most of what you'll see today was using acridine orange as the fluorescent dye. So the real application that we've focused on a lot over the last 5 years or so is in the area of imaging the inside the abdomen. So confocal microlaparoscope means laparoscopic surgery is when they go through a port in the abdomen and allow you to visualize what's going on in there and extract tissues out. You can do oophorectomies, hysterectomies. You can do appendicitis is mostly done laparoscopically, removing the appendix. There's lots of surgical procedures for which having a microscope capability in that context could work. So here's a device we've built. It's got that miniature lens on the end, a fiber bundle that runs through, a motor that moves that fiber to change the focus, another motor that drives a syringe, drives a small amount of dye through a hypodermic tubing out to the end to deliver dye, and motorized controls to control focus, dye delivery, and image capture and whatnot. So here's an example of that in the surgery suite. Jennifer showed a similar thing of the OCT system. She's working with Dr. Hatch as well, one of our collaborators. So here's our device. Here's what they see in a normal laparoscopic view. So here he's holding a camera, and he's visualizing what's happening. There's the ovary. It's placed in an endo bag. That's how they often will resect it and take it out through the same port that they're or another port that is inserted into the abdomen. And then in addition, our device is coming in and making contact with the ovary and then being able to visualize the cell distribution on the surface. Just one more view of that. Dr. Rouse over here by the instrumentation. So this is where the confocal microscope part of this is, the scanning system and the computer and image capture and all of that. And a fiber bundle runs over to the device. They hold that handle and do the imaging there and try to make a diagnosis in situ. So here's an example of a real-time in vivo. There was some dye delivery. Now it's getting placed onto the surface of the ovary and focusing. And you can see it moves around, but it's running at 30 frames a second, so you can get pretty good visualization of the distribution of cell nuclei as they're trying to hold that fairly steady, and it's not so easy to do. So that's one of the tricks, is to get a steady image, although you're continually collecting these images and can go back and review whatever you've collected over that region. So one of those frames looks something like this. What are we looking at? Well, the normal ovary has this epithelial layer of cells on the surface. So the nuclei are packed very close together, single layer thick, and that's what you're visualizing here. This is what's called an en-face view. The probe goes up in contact with the surface. So we're imaging this plane of the tissue, not the cross-sectional view that's being shown in the histology. When you get to a ovarian cancer, now that epithelial layer is significantly modified. It's a cancerous condition where the cells now on the surface are much more spread out. You have a thickened epithelial abnormal layer, and you can see quite a bit different visualization of the structure, the microstructure, architecture of the tissue as being an abnormal case. Just a few examples of what's normal and what's abnormal in terms of ovarian cancer. These are all different kinds of subtle structures, but you see this single layer epithelial, normal epithelium, closely packed cell nuclei, and then underlying that is the stroma. Sometimes that layer is denuded, and this is just a stromal layer. It's normal, but it's denuded, or the surface layer is gone. But you can see quite dramatically different view of what cancers look like. So this has been able to show, ex vivo, we've been able to show quite good sensitivity and specificity for detection of ovarian cancer with this technique, and they're now running an in vivo clinical trial of this instrument to see how well we can do things in vivo. So here's an image of a normal ovary in vivo, one frame out of a movie, and here's a high-grade serous adenocarcinoma of the ovary, so ovarian cancer. You can also do this in other organ sites. One of the big areas where they do endoscopy is in the esophagus. So there's a condition called Barrett's esophagus. It has nothing to do with Harry Barrett. It's the doctor that defined the condition decades ago. But you have the normal squamous epithelium on the surface of the esophagus, but people from gastric reflux and other problems can often get this condition where you see these glandular structures. This looks like the morphology or tissue type of the intestine rather than the esophagus. So it's called a metaplasia. It's a misplaced kind of tissue. And it is a precursor lesion for adenocarcinoma of the esophagus. So people who have this condition have to go through yearly, at least, usually biopsy procedures where they might take 20 pieces of tissue out of the esophagus and do all these biopsies to see if anything is changing from a normal condition or changing from the metaplasia to a dysplasia or a cancerous condition. So that's another application. Dr. Rouse and one of the graduate students, Xiao Wu, is working on trying to develop an improved flexible system that can go through the therapeutic channel of a standard endoscope to be able to image the esophagus. Along the way, they're developing a much better lens system that can accommodate a bigger fiber. This is 50,000 elements. And this lens is designed to be accommodated over a much broader range of wavelengths. You saw there were a lot of different fluorescent agents that we could use, and we wanted this lens to be able to work with many of them. So this is achromatically corrected from about 500 nanometers up above 800 nanometers, and it needs to be corrected over a range of conjugate positions. So it's quite a challenging lens design, and then really challenging to build it into a 3 millimeter diameter package. So here's an example of that lens, and here's an example of the nearly diffraction limited performance measured, the blue line, versus predicted performance of a diffraction limited lens, and an example image of imaging a slide with that. The yellow circle is the field of view that it was designed for. So very nice lens. Another area where we've been working is in the area of trying to image inside the fallopian tube. There is a belief that many of the origin of ovarian cancer is not directly on the epithelial layer of the ovary, but rather in the fallopian tube. So what we'd like to be able to do is get into the fallopian tube. And the way the ovary looks is it's something typically about that size, like a walnut, and it's got something about the size of this laser pointer as a fallopian tube coming close by it with this fimbriated end. So you've got this little thing coming off of here with our probe, our rigid probe, coming in with a laparoscope. If we came in, it would be hard. We can get into a contact with this, or we could potentially get into contact with some of that, but getting up into that tube and wrapping around could be quite a bit more difficult. So she's been working on, again, this is the work of Xiaoyu Wu, has been working on a fallopian tube flexible articulated tip system. So by tipping the handle relative to this rigid part, it will tip the thing back and forth. And maybe it's a little easier to see in this movie. So she's manipulating the position of this relative to the handle, and you can see the end is articulating in different directions. And at the end, you'll see she locks it into place. So if you're visualizing, you get it oriented where you can get into the fallopian tube, lock it into place, and then be able to insert it forward into the fallopian tube. So I want to talk just very briefly about a couple of areas that we've been trying to push this technology to improve upon it. I said we have a line scanning or slit scanning system. So this is a line that's scanned in one direction. The standard way that confocal microscopy is done is with a point that scans in a raster across the 2D image. That takes, as you could envision, takes quite a bit longer to do than a one-dimensional scan. So the advantage here was speed. The disadvantage is there's a loss in performance, especially in the axial resolution or thickness of that optical section. Typically, at least in terms of the parameters that we're using for numerical aperture, we're running with something about 25 microns of optical sectioning capability here, and with the same NA, we'd be able to be under 5 microns of axial resolution with a point scanning system. But it would be slow. So there are some approaches to parallelizing this from a point to not just parallelizing along a line, but parallelizing in an idea called the Nipkow disk. I won't explain this, but systems have been developed. There's a spinning disk that has holes in it, and you have to have matching sources and detectors, and it's pretty inefficient, but it does work, and it allows real-time imaging, and it improves upon performance of a slit scan. So we've been looking at another approach that, rather than having a full slit, has a set of points in there, and that will scan in a different way. So it's the same system exactly, except all we do is put in a multi-point mask. What this looks like is a bunch of apertures in here, and this thing spins very rapidly. So one way to think about how this works is, here's that mask. It's got a duty cycle of about 10%, so it's only about 10% open. So if you put that in front of the slit, you get a set of points that are separated. And if you now spin that thing, you can see that those will move through there, and the scan mirror in the other direction will scan the other direction of scanning. So as that thing would move through there, you'd see those moving up, and as it slowly would scan in the other direction with the scan mirror, it would cover the whole 2D field. So indeed, it works pretty nicely. In fact, here you can see what is with the slit scanning, with the multi-point scanning. The axial resolution improvement is exactly what you would predict, from about 25 microns full width half max down to about 4 or 5. or maybe even a little bit better than that. So, and you can also see it has these very long tails to the point spread functions, which is not such a good thing. So this is really a better image. What we're seeing here are the nuclei. This is a section of colon, and you can see the nuclei on the little microstructures on the surface. No, this is liver, right? Liver, yeah. This is a liver, and so it's the caniculae of the liver, and you can see the nuclei of the cells on that tissue. So one of the former students, Anthony Tambaguchi, who many of you might have known or met, did a very nice Monte Carlo study as well to show not only do we get the resolution advantage of the axial resolution improvement, but we can image in a scattering media like tissue much better. So this is a simulation of where do photons come from. If you're trying to focus down into here with a point scanning system, where do most of the photons come in a scattering media? You can see they come from the focus, but there's a lot of photons that come from other places that cause blur. And similarly, in a slit-scan system, you can see it's much worse. There's a lot of photons, a lot of locations that would give light, scattered light, into the image rather than just the point. And it turns out the linear array, this idea we've been working on, is almost as good as the point scan. So just to finish that concept off, here are plots of looking at how deep you're imaging, d times the scattering coefficient. So it's essentially numbers of mean free paths in scattering. So at about 1, you can see that this is the axial resolution plot. You can see that at about 1 scattering mean free path, you're going from 25 microns resolution to things getting really blurred out and essentially not even sectioning hardly anymore. So you can't go very deep. That's the problem in a scattering media. Whereas if you switch from slit to linear array, you recover almost the performance you would get out of a pinhole, both in the axial resolution and in the lateral resolution degradation of the system. So that's very nice, and we're continuing to try to push on incorporating that and getting a few bugs worked out of the spinning disk system. Another kind of advance or technique or idea that we had about using this system was to combine the confocal microendoscope idea, this NFOS view of tissue with a probe coming down, to the nice idea of OCT imaging to be able to image cross-sectionally through the tissue. And it occurred to us that you could do confocal imaging with exactly the same setup with some minor changes. Bring in, if you want to do OCT imaging, bring in the OCT light that's going to be reflected. So it's usually in the IR, about 800 nanometers or so. Do the exact same thing with a cylindrical lens and a regular lens to create a line illumination on the sample. And then with everything fixed, mirror fixed, bring that light over through the dichroic mirror. It'll go through that because it's longer wavelength. It'll image onto the slit. It'll go off of then scan mirror 2 and be dispersed. Instead of by the prism, we're using a grating. So go to a fold mirror grating and disperse the light once again. So this is an idea of how frequency domain OCT systems work. They disperse the light and take a Fourier transform. So let's just think very briefly about how that works. So here's a tissue sample. This is what's called a common path interferometer. So in most OCT systems, you see going out to an interferometer with a beam splitter, and there's a fiber going one way, another fiber going another way, and you interfere the beams coming back. This uses a common path where you use the reflection off the glass interface at the very tip of the lens, and then the tissue also has scattering or reflecting surfaces or locations that scatter or back reflect light back into the system. So these two beams will interfere, just like any interference experiment. There's an amplitude and a phase associated with both the reference and the sample arm of this interferometer. One has a slightly increased path length. That delta is the OPD, which is half of the z distance. So OPD is 2z. And so you get a sample arm and a reference arm. You combine those into a system and take the intensity. What do you get out? You get a constant plus an amplitude of a cosine modulation. And depending on how deep that is, this frequency of spatial period or period with respect to sigma, the wave number, will vary. So this is 2 pi wave number OPD. So it looks like that for a monochromatic wave. What happens with a polychromatic wave? Well, that is polychromatic, so it's just the same thing. You get some variation with wave number on amplitude and with that constant, but in general, this is a signal. If we ignore these kind of subtle effects of sigma, which affect axial resolution of the system, what happens when I Fourier transform it? If I take this signal and I disperse it with respect to sigma, take that grading and disperse it across a 1D detector, I'm measuring then on that detector as a function of sigma, and I take the Fourier transform with respect to sigma, I get, for a cosine, 2 delta functions. Jack Gaskell taught me this. Where is he? All right. I always have to get some Fourier in for Jack. You get 2 delta functions. You get something at DC corresponding to that, and you get an image. This one you ignore. That's the other side. On this side, at some increasing depth, from that scatterer, you got that image of the scatterer. So if you have multiple scatterers in here, you have a bunch of waves coming back, and you build up a whole Z-scan, or what's often called an A-scan in OCT, of the information in depth. So OPD is 2Z, so this is by just taking a Fourier, by dispersing onto a detector, taking the Fourier transform, you get a image along one beam of the light going through. So what is interesting about what we've done is by having a slit, you're getting all these parallel spatial versions of that. So this is after taking the Fourier transform of a object which had a few scattering planes in it that giving signal back to be able to measure what's at varying depths. And this is all done with no moving parts because it's parallelized along the slit. Each of these is the light is being illuminated. The light's going to the 2D detector. It's dispersed in one direction in the sigma or wave number space and spatial in this other direction here. So what's on the camera after you take a Fourier transform in the vertical direction is an OCT image. No moving parts. Nothing has to scan. And you can read this out as fast as your camera can go. So it looks great. It sort of produces what you'd expect in terms of at least seeing some highly reflective objects. You start going to biological or less reflective samples, you see there's some problems. It works, but signals drop off very rapidly. So what's going on? So Matt Reese and one of former students, Hussein Makhlouf, have looked at this quite extensively of why doesn't this FD-OCT system using a fiber bundle work as well as we expected. So the situation is that you have now a multi-mode fiber. Each of the individual cores in a fiber bundle supports multiple modes, not just one. It's not a single mode fiber. So you have illumination coming in. It's going to produce multiple waves coming out, multiple modes of the field. Each of those modes is going to reflect up and come back and produce multiple modes on transmission back through the fiber. So if you look at the reference field coming out, there's a sum over the modes of illumination and the modes of detection in what's produced when on the way back. So you get this for a reference signal. You get this for a sample signal with the delta still there. That's still the OPD, but the amplitudes and the key thing is that there's now phase differences between the modes. So each of these phases is indexed by IJ in both the reference and sample. So when you combine the signals, take the square magnitude to look at it in the intensity, now you get the same thing. You get cosines, and you get some constant, and you get some amplitude. Those aren't the problems. The problem are the phases. What happens when you add a bunch of phases of cosines? Cosines are the same frequency, but different phases. Tend to cancel out, right? So these will tend to cancel out, and we get less sensitivity. So we did some experimental work to show that this is what you would predict for a perfect OCT system with the blur function of the size of the fiber element, and basically any blur that you get on in terms of the detection in wave number is going to blur out the higher frequencies, those higher modulation frequencies, which correspond to greater depth. So there's a natural fall-off in all FD-OCT systems that have to do with that blurring. But in addition, because of all of this loss of the modulation signals that we're looking for due to the various phases, we get a reduced sensitivity that is causing the basic problem. Moreover, this is even worse than just a fall off. If this comes down to 0.4, you'd expect that curve to sort of follow a reduced version of this. But it falls off even more rapidly than that because the parallelized version of the fiber bundle has multiple fibers in there that are, once you go deeper, more and more fiber illuminations are overlapping. So you're getting another summation over multiple fibers within the bundle. So we think we have a handle on this, and the solution is really to come up with a single-mode fiber bundle for this application. Okay, so with that, let me shift gears a little bit and now talk about another project that is in the area of multimodality imaging of window chambers. So completely different shift of gears. Any questions on the previous part of that? Clear as mud? Okay, so what are window chambers? Here's an example of a type of window chamber called a dorsal skin fold window chamber. So it's mounted onto the dorsal skin fold. Usually the skin is removed. This is put in place with a cover slip over it so that you can get access to visualize this optically. So it's implanted into the dorsal skin fold. The top layer of the skin is removed, and a cover slip is placed over the top of it. So this is a technology that's been around for a long time that people have been using to look at cancer biology, vascular biology, lots of things. Here's a nice example of the quality of the images that you can get. So this is a dorsal skin fold window chamber where some cells that are expressing GFP. So you can transfect cancer cells to express a green fluorescent protein, like jellyfish. And if you shine the appropriate light on them, they'll fluoresce. So this is a combination image that is transilluminated, so you can see this thin layer of tissue with all the vessels flowing through it. And then you can see in another kind of wavelength, slightly different wavelength, you can see the fluorescent light here. So this is fluorescent tumor, and this is normal tissue surrounding. And you can see the vascular supply. This thing is starting to grow. I don't remember exactly the date. It's probably a week after implantation of the cells. At 2 or 3 weeks after implantation of the cells, the tumor has grown, and you see all these microvessels coming in, very chaotic, abnormal vessels that are associated with cancer. So this is a fantastic model. You can visualize at very high resolution what's going on in very much detail, the vascular supply, how the tumor is growing, things like that. One of the areas that we've focused on in using this has been in the area of trying to image the pH distribution. It's well known that tumors are acidic, and they create an acidic environment around them that leads to their survival relative to normal tissues. In other words, they're able to make an acidic environment in which they can live well and normal tissue dies off, which allows them to invade into surrounding tissues. So one of the graduate students, Wei-Min Liang, has been working on using this approach to measure pH in tumors. And so we've used this SNARF1 dye, which has fluorescent properties. When it's protonated and deprotonated, it changes its spectral emission properties. Its fluorescence emission changes as a function of pH. So if you take a measurement in 2 different spectral bands, you can come up with a ratio of 2 signals that is monotonic with pH. And so with calibration, you can convert a measured ratio into a pH. So the way this works, here's another example, GFP tumor. So a tumor that's expressing the GF green fluorescent protein. It's illuminated with a laser light to make it fluorescence. Turn that laser off and go into the excitation wavelength that's different for the SNARF. Here there's no SNARF. We get a background signal, add SNARF signal, fluorescence increases, background subtract, and then take the ratio and calibrate that to generate a pH map. And you can see very nicely that the GFP tumor is darker, meaning more acidic. And in fact, it is making its surrounding areas even darker as well. So this would be an example one might predict that this tumor would tend to grow in this direction by creating something that is a potentially more acidic environment in that direction. So that's been very nice work that we've been working with some colleagues over at the Cancer Center on this. Is that for me? So one other area that we've become very interested in is in the area of breast cancer. So Wei Min and Rachel Schaefer in the BME program have been working on developing and utilizing a model of for breast cancer. So this is a different model where we create a plastic structure that can be inserted over the mammary fat pad, and we can grow what's called an orthotopic tumor, one that's in its correct place. So we can insert human, this is a SCID mouse, so it's severely immune compromised mouse that is able to support human growth of a human cell line. And so this is implanted under the skin, or the skin is taken away and this is implanted in there. So we have, again, the window chamber on the inside to be able to visualize the mammary gland. So this was built with the Connex 3D printer system that's over in the radiology research area that the CGRI people help support. So it's a nice model system that now allows direct visualization of what's going on inside the breast. And so here's just a white light illumination. You can see some little microvessels, but where's the tumor? Almost impossible to tell. But since we're using the GFP labeled tumors, transfected tumor line, you can see very nicely where the tumor is in here at low resolution. And you can push up to even much higher resolution. So this is a 40x objective and see individual cancer cells. So these are GFP cancer cells, very nicely defined. In this case, we injected then another dye at a longer wavelength, Lexaflor 647, into the vascular system, fills the capillary, so you can see a perfect registration of what's going on between the capillary bed and the cells that are surrounding it. So all the way from the sort of macro visualization of that previous slide to the microscopic, cellular, and even molecular level imaging capability that you can get out using labeled fluorophores. Here's a nice example, again, at the kind of macro scale of injecting that same, what we saw before. This is just this kind of situation, except now looking at the whole thing. So you inject the contrast agent, you see it fill all the vasculature. It's filling the microvasculature within there as well. So this is something called dynamic contrast enhanced imaging that can allow you to look at the vascular properties of the tumor. Tumors generally have very poorly formed vasculature. They're leaky. They don't connect properly. And so one of the problems with chemotherapy and other kinds of therapy is that the drug never gets there because the vascular system is so poorly formed. And so that's part of the reason why these things become hypoxic and part of the reason why they become acidic. So there's this whole theory of what's going on in terms of the microenvironment of tumors that this technology will allow us to study very nicely. One more example of high-resolution imaging at the molecular level. This is CA9, which is a particular molecule that's associated with the transport of protons across the cell membrane. So usually in cancer cells, CA9 is upregulated, and in that case, you can see the cells, the orangish cells are the ones that are expressing a lot of CA9 on their surface. So again, consistent with this highly acidic environment where the cells are actively trying to pump protons out of the cells to stay alive into the microenvironment. One more example. Here's an example of imaging, again, related to this whole story about hypoxia and pH and acidity and whatnot, is trying to directly image hypoxia, meaning where are the tumors getting insufficient oxygen. So this is the work of Rachel Schaefer, who's been trying to develop a way of imaging hypoxia, and she's come up with a very clever way of coating a material, platinum porphyrin, in polystyrene on the surface of the cover slip. That has a property that its phosphorescence lifetime is dependent on the local oxygen. Oxygen is a quencher. It makes the excited fluorescence state decay more rapidly. Turns out this is a long-lived material, so this is in the microseconds rather than nanosecond range of fluorescence lifetime, so it allows the use of imaging instrumentation. It's a little bit easier to use for measuring fluorescent lifetime. But the point is that in areas where there's a lot of oxygen, then you're going to decrease the fluorescence lifetime. So in this area out here, which has a long fluorescent lifetime, there's not much oxygen. So she's overlaid the vascular image on top of what she got out of the hypoxia image, this lifetime image, and you can see that the vessels run along areas where, in general, this appears to be areas that are well perfused, and there's an area out here that's very poorly perfused and is probably hypoxic. This is pretty preliminary early work, but it's very promising in terms of being able to directly image hypoxia. Just one more example of this is in the mammary window chamber of being able to image pH as well, the same kind of thing. Wei Min's been looking at trying to modulate tumors' pH by sticking in things like MIBG makes them more acidic. You can give the animals bicarb, make them more alkaline, and increase the pH. And that's actually being investigated as a potential therapeutic approach to alkalinize people to make them less sensitive to tumor growth. So the one thing about this that's really nice was that this was a plastic window chamber. This is made with that printer system that's a photo polymer, and so it's essentially MR compatible. So we can uniquely, in this plastic window chamber system, do multimodality imaging. Not only optical imaging, but take it in and do MR imaging. So here's a setup where there's a surface coil on here. It's going into a system. Now we're using a volume coil in the system for excite and a surface coil for receive. But you can see the whole setup. There's gas anesthesia. There's various respiratory monitoring, temperature monitoring. A lot of things have to go on to stick an animal to be able to keep it alive and warm and happy in the magnet. But you can do it, and when you do it, you get very nice images of tumors. So this is a cross-sectional view, sagittal view. Here's the window chamber structure along there, and this is the mammary fat pad, the lining, the abdominal lining, and you can see there's a tumor that's been implanted and growing in very nicely on the surface. And here's the sort of en-face view. This would be like the confocal microendoscope view of sticking down on top of the window chamber. So there's the window chamber, and you see the tumor there. So another example, this was over a period of a couple weeks where we implanted, the tumor started to grow. This is about 1 week out, and here's the fat. The fat in MR images, or at least in T1-weighted MR images, is bright. So the mammary fat pad is the bright signal, and you can see the infiltration of this tumor into regions of the fat pad. So this is tumor here, there's probably tumor there, and you can get a better sense of it a week later that it's completely, this whole mass here has completely filled and moved the fat out of the way and overtaken this area. There's a technique in MR that you can inject a contrast agent called gadolinium DTPA, which will shorten the T1 and increase the signal intensity within the tumor. And it goes preferentially into the tumor, and so with injection, this tumor injected with GAD, we can very nicely see this tumor. You can see that it's growing quite deep into the tumor. This is about a centimeter across here, so this is probably about an 8 millimeter or 6 millimeter tumor in size, so it's growing pretty big. So this is a way of looking at how a tumor will grow over time, and then being able to optically look at the very high-resolution molecular events that are occurring at least in a limited portion of the tissue near the surface. Another thing that you can do with MR has all these fantastic capabilities to be able to look at physiology and make important kinds of measurements that are useful for diagnosis. One of the things that diffusion MRI is a very important kind of approach. It's used when somebody has a stroke, they'll almost certainly go in and have a diffusion MRI. What happens in stroke, cells swell, water goes in and is restricted. So the diffusion properties of water become restricted, and it gives you a bright signal. So in a diffusion-weighted image, if something's highly cellular, like a tumor or swelled cells that are ischemic from a stroke, you'll get a bright signal. So you can see very nicely a bright signal within the tumor. The nice thing about this is that you can do this measurement of a diffusion decay of signal with varying what's called B values, weightings, and so you get I guess from your perspective, a decreasing signal. You can fit an exponential decay and come up with a quantitative measure, the ADC, the diffusion coefficient, or apparent diffusion coefficient, MAP. And this is a quantitative value for how much can water move around, how rapidly, how many microns per, it's sort of a variance, how many microns squared per milliseconds do the things move around in the tissue. And this is very useful from the cancer standpoint of a very sensitive biomarker for looking at response to chemotherapy. Turns out that you'll see changes in the diffusion coefficient in a tumor well before you'll start to see any changes in size. So if you can go in and do an ADC test on a patient and see that they're getting chemotherapy and their ADC has changed, becoming increasing, then you could tell that they're responding to chemotherapy. You keep them on it. If they're not, you can change the therapy and modify something before giving them a long regimen of chemotherapeutic drugs. So it's a very important area. The last imaging technique I'd like to talk about just very briefly is, besides MRI, there's a very clever way that the CGRI group, and I've been collaborating with them on this, of developing a way of imaging, doing nuclear imaging. So many of you know that Harry Barrett's group does a lot of, develops all kinds of sophisticated imaging instruments to look at radio tracers. You inject something that's radioactive, the emission of gamma rays or positron emission will create a visible or a signal that can be detected, and you can image that distribution. So, for example, one of the classic ways of doing imaging in cancer is with what's called a PET scanner. You have a radio tracer, which is FDG. It's basically a glucose analog. Tumors are very highly metabolic. They take up a lot of glucose to stay alive. And so they're going to get a lot of FDG in them. And it's a positron emitter. It's something that a positron annihilates with an electron, 2 counterpropagating gamma rays. You can detect that with coincidence counting detectors and have a whole array of these detectors and do PET imaging. And it's sort of the classic way, one of the classic ways of looking for metastatic spread of cancer in patients. It's a clinical tool. It's used all the time. But it's a very expensive and complicated instrument, and it also is low resolution. So it turns out in the window chamber, because the tumor is so close to the surface, that if a tumor takes up the FDG, it can emit a beta particle, it's a positron, will be emitted, and it's so short distance that it can actually escape. It doesn't annihilate with an electron, at least some probability that it can escape. And so you can stick a scintillator material on the top of the window chamber, and now the positron will interact with the scintillator and create light. And so you can image the light onto a very low light level camera. This is kind of like a bioluminescence box. There's a lot of these out there for doing basic biology of looking at weak fluorescent signals and whatnot. So this is just a simple system, a low light level CCD, imaging system, lenses, and the FDG injection. And what you get out is something that is what the red is the color code for the FDG signal that we got in this low light level camera. We can open up the box door a little bit and just see the whole visualization of this thing. We can put a little bit of green laser light on this and make it fluorescent. So this is co-registered image of where the tumor is in GFP, and then this is the overlaid FDG image, and you can see a very good correlation between the uptake of the glucose where the tumor is to the signal there. You also see this other signal coming out here and extending out a little bit. If you remember, how you do this is you cut away a little bit of skin. So there's potentially an inflammatory process here, which is one of the confounding things in PET imaging. When people have inflammation, it's one thing that can give you a false positive PET result that it's inflammation and not cancer. So we believe this is an inflammatory response at the surgical margin of the tissue. So this is just a few examples of the multimodality kinds of imaging things that you can do with this window chamber. And I think it's really a very powerful technology that will allow us to look at tumor growth, angiogenesis, all the vascular properties of the of the tissue, look at things like pH and all of the associated things associated with hypoxia and molecular processes of why tumors are so acidic, and can you use that to advantage to make more effective drugs. Vascular permeability, metabolic activity, the glucose consumption of FDG, receptors, molecular pathways, hypoxia, all these things I think are doable, all in the same setup, in the same co-registered ability to look at how things relate to one another. So with that, let me just end by saying this is really the work of a lot of people, as always. I have some very good collaborators over in the medical center that have helped, especially in the area of the confocal microendoscope project. As you can see, that's in the clinic. And so we have Dr. Chambers and Dr. Hatch are the surgeons that we're working with, that are willing to get in there and use our instrument inside human subjects. And I thank them and applaud them for their expertise in helping us with that. Zhonglin Liu and CGRI has helped us with the gamma imaging, the positron emission beta imager, we call it. Wenxin Zhang is a pathologist that's helped us. And a lot of graduate students have worked on this over the years. I mentioned the ones that are currently in the lab: Wei-Min Liang, Matt Risi, Rachel Schaefer, and Xiaoyu Wu. But a lot of former students have contributed to this work as well. And finally, let me thank the people that helped us with animals, the Center for Gamma Ray Imaging 3D printer. Cecile Shea is one of the people that's done a lot of the work of running that and getting us parts for the window chamber parts, et cetera, and of course funding. So thank you very much.